Now let's talk about the actual process of digestion, which is why we have a digestive system to begin with. So we'll start off with the digestion of carbohydrates, where again we eat a carb, like polysaccharide, and we start breaking down the polysaccharide through amylases into maltose. We can also ingest disaccharides like sucrose or lactose, and those are digested into their monosaccharides by the enzymes sucrase and lactase that are found within the intestinal brush border into their monosaccharides. Maltose is further digested by maltase enzyme, and all three of your monosaccharides, glucose, fructose, and galactose, are what, again, the intestinal cells can absorb. So we'll take a look at this in more detail. So the digestion of carbohydrates starts within the oral cavity by the help of the salivary amylase. But once we swallow that bolus, salivary amylase is deactivated by the hydrochloric acid found in the stomach. Nothing really happens to carbs in the stomach. It's not until the carbs moves on to the small intestine where it will be acted upon by pancreatic amylase. Remember, any amylase is going to break down your polysaccharide into the disaccharide maltose. Now, for the two other disaccharides, sucrose and lactose, we actually ingest them. So sucrose is your table sugar, lactose is your milk sugar, and we will break down now all three of those disaccharides, maltose, sucrose, and lactose, by the enzymes again, maltase, sucrase, and lactase, into your three monosaccharides. And then those are going to be absorbed by the small intestinal epithelial cells that we call your brush border. The monosaccharides can be transported across the intestinal epithelium into the bloodstream, and then from the bloodstream through the portal circulation over to the liver. Most of the ingested carbs are digested and absorbed within the first 20% of your small intestine. So we'll take a look at how the digestion occurs, and especially the absorption process. So what are we looking at here? We're looking at the intestinal epithelial cell. This is the lumen of the intestine that has all of the goodies that were just digested and that we need to absorb. This means that this is the apical border of the cell, while this is the interstitial fluid that would have the blood vessels. So this would be your basolateral membrane of the cell. Now, in order for to absorb these monosaccharides, it would first have to pass through the apical border and then through the basolateral membrane. So let's see how that occurs. For fructose, passes the apical membrane through facilitated diffusion on a glucose transporter. GLUT stands for a glucose transporter. As for glucose and galactose, those are co-transported with sodium on this active transporter known as SGLT, or sodium glucose co-transporter. Now, all three of your monosaccharides are going to go through a glucose transporter in order to exit the cell through the basolateral membrane into the interstitial fluid and then off to the venous circulation, and all of that will be shuttled over to the liver. Now for your proteins, proteins, we intake them as polypeptide chains, and the first step was to break it down into smaller peptide chains, and that would be through the action of pepsin, trypsin, and chymotrypsin. And then these little fragments, we would want to remove the carboxyl group and the amino group, and we do that through carboxypeptidase and aminopeptidase. We end up having these amino acids that we are able to absorb. For the peptide fragment, you can find here the amino acid would have an amino group on one side and a carboxyl group on the other side. And that is why you want to remove these groups in order to absorb that amino acid. So we are going to remove the carboxyl group by an enzyme known as carboxypeptidase that is secreted by the pancreas. And we'll remove the amino group through an enzyme known as aminopeptidase or aminopeptidase that is found in the brush border. So protein digestion starts in the stomach. Nothing happens to proteins when we in the oral cavity except for chewing. But it's not until we reach the stomach where protein digestion is going to start by the action of pepsin. And remember, pepsin is released as pepsinogen that is activated by hydrochloric acid. And then when the peptide fragments move over to the small intestine, we have the enzymes trypsin and chymotrypsin that are going to further break down the protein, and those two enzymes come out of the pancreas. And then we have to break out those carboxyl and amino groups, and that is through carboxypeptidase that is released or secreted by the pancreas, and the aminopeptidase that is found on the apical membrane of your small intestinal cells. In order to absorb, again, it has to go through apical membrane, through the cell, and then over to the basolateral membrane. So at the apical membrane, your free amino acids are going to enter either by secondary active transport with sodium or secondary active transport coupled to hydrogen. On the basolateral membrane side, they are going to be transported through facilitated diffusion using an amino acid transporter. So we'll see what we've been talking about. This again is your small intestinal cell. Here is the lumen of the intestine that has the chyme and has the amino acids that we want to absorb. At this point, we have already digested them into amino acids. So you can see here, part of the amino acids are going to be absorbed, transported or co-transported with sodium, while some small peptides are going to be able to be absorbed here through being co-transported with hydrogen. But again, these small peptides, we definitely have to break them down using peptidases that are found within the intestinal cell into these smaller amino acids. And it's these little monomers that can go through these amino acid transporters into the portal circulation and then being shuttled off to the liver. Remember, anything that is absorbed, all of that has to go to the liver first through the portal circulation. Now moving on to triglycerides. Remember, we've got a glycerol and the 3 fatty acid chains. It's all up to the lipase to break it down into a monoglyceride and 2 free fatty acid chains. The absorption, though, of triglyceride is a little bit more detailed, though. So fats digestion starts in the oral cavity through salivary lipase, and then a little bit in the stomach through gastric lipase. But by far the vast majority of digestion of fat happens in the intestine through the action of pancreatic lipase. It is also helped with bile. We definitely need bile in order to do that. And all of these lipases split the triglycerides into monoglyceride and 2 fatty acid chains. Before these enzymes are able to act, we have to break down those big fat globules into smaller ones. And that is a process known as emulsification. And that is helped by bile salts that are found in bile and with the help of phospholipids, together with the movement of the small intestine. So as the small intestine moves, it'll break down those bigger fat globules into smaller ones. And after absorption of the monoglyceride and the fatty acid chains, the intestinal cell is going to reconstruct them back into triglycerides. And then that triglyceride will be absorbed into the lacteals, which are the lymphatic capillaries found inside of the intestines, and they are going to be transported as chylomicron. Chylomicron is a triglyceride surrounded with proteins. Remember, triglycerides are fat, so they are not water soluble. In order to be transported, they have to be surrounded by proteins. So here you can see what we want to do. So this is the fat globule that we ate. In order to break it down, in order to digest it, we first have to do what is known as emulsification, which is breaking down this fat globule into smaller droplets and surrounding them, coating them with bile salts and phospholipids. Those are going to help the enzymes work because it makes it easier. Instead of working on this very huge flat globule, now the enzymes can digest these smaller droplets. Now here again, you can see the whole process where you got this fat droplet, breaking it down using the bile salts and phospholipids into these little droplets. And then the pancreatic lipase, or all of the different lipases, are going to digest the triglyceride into these monoglycerides and these free fatty acid chains. It'll go in through the intestinal cell. In there, it will reconstruct the triglyceride again. So the smooth endoplasmic reticulum of the intestinal cell is going to take a monoglyceride and 2 free fatty acid chains and make it into a triglyceride again. Again, triglyceride is just a fancy term for fat. So now we're back to a whole fat drop molecule, which cannot be transported in, is not soluble in water. So you see it being coated with those little red protein dots in order to make it water soluble. And instead of being absorbed like our monosaccharides and amino acids into the bloodstream, these chylomicrons go into the lymphatics of the small intestine. These lymphatics have a very fancy name. They're known as lacteals. For the absorption of vitamins, well, we've got 2 different kinds of vitamins. If it is fat-soluble vitamins, these are the vitamins that can dissolve in fat. Those are vitamins A, D, E, and K. Those follow the pathway for fat absorption. While your water-soluble vitamins, which are the rest, okay, those are absorbed by either diffusion or mediated transport, with one exception, and that is vitamin B12. Remember when we were talking about the stomach, we said that the stomach makes intrinsic factor. And intrinsic factor, we mentioned, was very important for the absorption of vitamin B12. B12 will then be attached to this intrinsic factor and absorbed through endocytosis at the terminal ileum, which is the very end portion of the small intestine. So if somebody has an autoimmune disease, okay, known as pernicious anemia, where the body makes antibodies against the parietal cells, that means that they've lost that ability to make intrinsic factor. So now they cannot absorb vitamin B12, and that can lead to that pernicious anemia that we talked about. And pernicious means that it's a very stubborn kind of anemia to treat, which is true. And these RBCs are huge, and that's why it's also known as megaloblastic anemia. Another cause for pernicious anemia would be gastrectomy. If you just surgically remove the stomach, say due to cancer, again, they've lost the parietal cells, no intrinsic factor, no B12 will be absorbed, and B12 is needed in order to make healthy red blood cells. Now, celiac disease is another autoimmune disease, and where we're making autoantibodies against the brush borders of the intestine, and that is going to be induced by the ingestion of gluten. For the absorption in waters and minerals, minerals are absorbed by diffusion in the small intestine, and water is going to follow by osmosis. So it's a very simple kind of absorption. Absorption of sodium, chloride, potassium, and calcium are really important to maintain a lot of physiological processes that we have already talked about. So bone health, nerve health of the nervous tissue, action potential, muscular contraction, all of that, we've seen how these ions are important. Iron is also important, but it has a more interesting stuff to talk about. So when iron is actively transported into the intestinal cell, it is going to bind to ferritin. Ferritin is a protein found inside of the intestinal cell. It binds iron. So remember that iron, the formula for iron is Fe. So ferritin means that this is a protein that attaches to iron. And the intestinal cells, or the enterocytes, are going to store iron inside of them. Just in case we ever need iron, let's say somebody bleeds profusely and they want to now produce a lot of red blood cells, well, they have iron stored in their enterocytes attached to ferritin. Hey, some iron, though, is not going to be captured by that ferritin and will enter into the bloodstream, But again, in the bloodstream, it cannot just move along on its own. It has to be attached to another protein. The protein that it attaches to in blood is known as transferrin. So transferrin is a protein that transports iron within the circulation. If there is a genetic defect in iron homeostasis, where there is an accumulation of iron within the tissues, that is a disease known as hemochromatosis. The treatment would be frequent phlebotomies, and which means that they would donate blood frequently in order to get rid of that extra iron. Now, so we talked a lot about the intestinal, secretions. Now let's talk digestion and absorption. Let's talk about intestinal movement. So there are two different kinds of movements. There's segmentation and peristalsis. Segmentation is where you'll find the part of the intestine that contracts and the neighboring part relaxes, and there's no actual movement. It's done to break down those bigger particles into smaller ones. So it's kind of like a churning movement where the chyme keeps on going back and forth in order to, again, break it down into smaller parts so that the enzymes have access to everything. So these are, the intestines contract and relax in these rhythmic patterns, but without peristaltic waves. So there is no actual movement of the chyme within the intestine. This rhythmic contractions usually occur during a meal, when, kind of like when you're trying to digest the food. We also have what are known as migrating myoelectrical complexes, or MMC. And this usually happens after a meal has been absorbed. Now we only have the waste products within the intestine. These are usually happen in order to kind of squeeze everything out of the small intestine to move on to the large intestine. So after digestion, absorption happen, whatever is left in the small intestine are just waste products. Now we want to get rid of it. And we'll do that through these very strong or, repeated waves of peristaltic activity. And those are known as your MMCs. These MMCs are going to start at the very beginning of the small intestine, and it would travel about 2 feet along, and then it dies out. And then another wave starts, which, kind of starts a little bit further down where the first one did. And it keeps on migrating down the small intestine. It'll take about 2 hours before it goes through all of the small intestines and reaches the large intestine. So now these MMCs are responsible for moving all of that undigested material, pretty much squeezing everything into and cleansing it, making it, pretty empty, pushing everything into the large intestine. This also prevents bacterial overgrowth, because if things remain in the small intestine, that would be a great medium for bacteria to grow. And this ends this portion.